In this video, we explain receptor editing. Which cells does it concern? It concerns B cells, in particular the immature B cells. Where are these cells found? They are found initially in the bone marrow. So we are in the bone marrow. Receptor editing will take place during B cell development. And remember, this is part of the central tolerance process. What do we see in the first panel? In the first panel, you see an immature B cell. This cell expresses an autoreactive receptor, this IgM. This is in particular this is a surrogate IgM, SIGM, okay, which is part of the B cell receptor at this phase. And here are self-antigens of one of the cells in the bone marrow, so self-antigens. The immature B cells now, in this case, express an autoreactive receptor, which recognizes a multivalent self-antigen. However, this B cell can further be rescued by gene rearrangement that will happen, and that will replace the autoreactive receptor with a new receptor that is not self-reactive. This is what we call receptor editing. So first of all, as we've said, the B cell produces the IgM, and at that point, RAG proteins, RAG proteins are still produced. Now, RAG proteins play a huge role in V and J recombination in gene rearrangement. We'll explain this in another video about the specific role of RAG proteins. Keep that for now. They play a role in recombination of the gene segments. If the receptor is not self-reactive, the absence of surrogate IgM cross-linking allows gene rearrangement to cease, and B cell development continues with RAG proteins eventually disappearing. But in this case, where we've got an autoreactive receptor, the encounter with the self-antigens results in what you see here, bringing the B cell receptors together as they bind to the self-antigens, and this is called cross-linking. Because of that cross-linking, RAG expression continues, and light chain rearrangement can continue. So gene rearrangement continues. In the second panel, you see the gene segments for the light chain. Now, these secondary rearrangements can rescue the immature self-reactive B cells by deleting the self-autoreactive receptor and replacing it with another sequence. Now, if the new light chain is not autoreactive, we'll see that the B cell continues normal development. If the receptor remains autoreactive, like in this case, there's still cross-linking present. The rearrangement continues until a non-autoreactive receptor is produced, or until no additional light chain V and J gene segments are available for recombination. So on the left, cross-linking is present, no further cross-linking, no more rag protein, no further gene rearrangement, and development continues. The importance of receptor editing as a mechanism of tolerance is well established, as defects in this process contribute to the human autoimmune diseases, systemic lupus erythematosus, and rheumatoid arthritis, two diseases characterized by high levels of autoreactive antibodies.